I've been working on making some graphics of my favorite protein, Argonaut, using this software called PyMol, which is free, and I want to help show you how you can use it to look at all sorts of different protein structures. And you can also play around with how you color them, how you represent them, so like sticks, ribbons, colors, all of that sort of thing. And then you can ray trace them so you get these really nice high quality figures that you can then use. And so it's this really cool tool, and so this isn't going to be like a full tutorial, but more just like an introduction, some of the main things to look at, keep in mind, to really just help get you a little familiar and comfortable using it to play around with some protein structures. So let's take a look. So our goal for today is to make this figure of Argonaut, AGO. So I've told you about AGO before. It's this protein I study that's involved in this process called RNA interference, which is involved in regulating messenger RNA levels, which is involved in regulating protein levels. And so I have videos and posts and all sorts of things on AGO. And yeah, so I was making graphics of AGO today for our presentation, and so I thought I'd show you how I do it. And so if you want to learn more about Argonaut, go to those posts that I'll link to, but I'm not going to worry about that for now. And so I was making these with software called PyMol. So this, what it's showing you on the screen, is not PyMol. This is another option I just wanted to tell you about, is this software called ChimeraX. It really is especially popular with like cryo-electron microscopy, so cryo-EM people. PyMol tends to be used more with like crystallography and stuff, but people use both for both. And so I said before, like PyMol is free, so that's kind of like with an asterisk. So there's different versions of PyMol. So the like full PyMol, like the license or incentive version, it is free for like students and free for teachers. So you can get like a license like that. You can also, like, labs will buy a license for their lab or whatever, and have it that way. If you just install it, but you don't have a license, you can basically do everything, except that if you try to like export a figure, it's going to have a watermark on it. But there's an alternative. There is a free open source version that so there's a free open source version which has almost all of the capabilities. Doesn't have some of the advanced capabilities, and it's a little slower, but so I'll put links to all of that. But I just also wanted to tell you about ChimeraX, which is also free, and is also an alternative. And there's also something called CCP4 molecular graphics that I haven't used much, but it is also an option that I will link to. So let's get started. PyMol isn't the most intuitive software to use, but there's a really great resource, the PyMol wiki online. They have tutorials, as well as you can search for anything. Basically, if you Google a question like PyMol something or another, it'll normally take you here. And they have a lot of helpful things like tutorials and cheat sheets and that sort of thing. So once you've downloaded PyMol, then you can just open it. PyMol window looks a bit like this. So you have these different areas. So like over here, this is like the command line where you can write in code, and it'll show up here. There's some like buttons up here. Over here you have a more like graphical user interface, and we'll have things show up here later. Over here, and here is where the like protein is going to show up. And so there are multiple ways that you can get like a protein structure. And so if the structure that I'm using is this, I have it memorized because I used it a lot. It's the structure of Argonaut bound to microRNA 20, which was solved by a lot of cryo-EM in our lab a number of years back. And so if you don't know, so the PDB code is like this four letter number code thing that corresponds to a protein structure. And so I have another posting video and stuff on the PDB numbers and nomenclature and how you can access these. But basically, even if you don't know the like name of a protein or whatever, so you can search for different terms. So maybe you want to look at their DNA polymerase, and so you search that. You can find like a number of different structures, and then each of them will have different codes that you can then put into PyMol. But I'm going to use the 4F3 key. So I can get it if I go to file, get PDB. I can do it this way. So it also gives you the option if you want to download the map. So it's a crystal structure, so it has like electron density maps, so more on that on crystal structures, but basically that's like the meshy thing that you put the like the structure into, so that you put the model into. So what we're going to see is the model. That's where the position of the different atoms, which is figured out using based on the location of the electrons around the atoms, is that how they interact with the X-rays. And so I'm not going to go into that here, but that's what these map things are. There's also chains, so you can have multiple chains. So if sometimes there are different proteins within the structure. Sometimes they're like proteins, RNA, other molecules like binding partners and that sort of thing. Assembly has to do with like sort of when there's like multiple copies of things in the PDB file, and so you can see like the chains and assemblies in the PDB. But if you just wanted to fetch like certain chains, you could do that here. Another way to get the PDB file is fetch. So I'm just going to put fetch for F3T, and that's going to give me that structure. Okay, so now we have this bright green thing that doesn't really look like that picture that we're going after, but we're going to get there, don't worry. Okay, so what you can see is we have Argonaut in green, and then we have that guide RNA, so the microRNA is in that weird color, whatever. The first thing we'll do is show you how to see the sequence. So you can do that by pressing this S button down here, or you can go to display sequence. So right now you'll see that we have the protein, and then you have this, so this is the RNA, and so this is actually as like a segment instead of a chain. So that was A, this is B, and then we have C and D, which are these phenol molecules over here. So it's just a small molecule it's bound to in the structure. And then all of this, these are these weird red dots that we see that are water. So those aren't very helpful. They're kind of distracting. So what we can do is we can either remove them or we can hide them. So there are a couple ways if you want to select something. So you can come and you can like drag it over here. Now you have this selection like over here, or you can refer to it as Sully. If you go to action, let's see what you can do with these different things. So you'll see that the 4F3T, if we click this, we hide everything. There's this ASHLC. So basically, the A is the action tab. So this allows us to do actiony things, and so we want to remove those atoms. Now we got rid of them. Let's say for the rest of this water, we can also do like select. So this is going to be select // E, because the // is just saying, because it's in 4F3T, and then the segment is E. And so if we want to hide this, so we can go to selection, hide everything, And now you can't see those waters, which is nice. But they're still there. We haven't removed them. But now let's try playing with some of those other things around there. So the S is show, which is more interesting than hide. So we have different ways we can show the protein and the RNA, and we could also do that if we go to like display. So there are tons of different options for basically everything, and a lot of the things you can do in multiple ways. So you can do by typing it into the command line. You could do using this chart up here, or you could do using the buttons on the side. So one of the easiest things to do or to play around with this when you're getting started is to try out the different representations. So right now we're showing this in cartoon mode, where you see like the alpha helices are in those like curly ribbons, and then the beta strands, so there's like arrows and that sort of thing. So this is the cartoon view. We can also set it to show in different forms. So if I put ribbon, but right now the cartoon's still showing, so I want to hide the cartoon so you can just see the ribbons. And so now it's just showing like the backbone of the protein. So let's look at a couple other ways we can display it. So if we want more detail about like side chains and things, we can show we're going to show this now as sticks, and we'll hide the cartoons. And so now you can see more of like the actual like side chains, like the parts sticking off of that main chain. And the coloring right now is in like atom mode, so you can see that the nitrogens and stuff are in blue, the oxygens in red, carbons in green. If you want to change the coloring, so we can also do it like by chain. So now you can see that the RNA is one color and the protein is one color. Sometimes it's nice to do a spectrum where you have it like in order, so going from like N terminus to C terminus, so you get like a rainbow around. But right now it's kind of hard to see it with the stick mode, so let's turn cartoon mode back on. So now you have cartoon mode and stick mode, which isn't very helpful, so we're just going to hide the sticks. And so now we can see Argonaut colored like that. So right now it's also coloring the RNA, but we'll change that later. What if we want to? We can also show the side chain as sticks. So now we have the side chain sticking off of the cartoon ribbon. Sometimes if you do this, it will give you weird things. So do is display or setting cartoon. So here's just like different options for how you want to display things, like how you want the loops to be and that sort of thing. But this side chain helper, so I have this on now, and you see it makes it now the sticks are sticking out better, nicer than with when you have it off. You can get kind of like weird stuff happening when you have the side chains. So sometimes it's a lot more obvious than other places. But we'll just put that back on. Okay, so now we'll hide those side chains. And let's go ahead and try to name something. So we want to let's separate that RNA. So if we go back and we color it by chain, okay, so now we can see that we have that protein is in green and the RNA is in blue. So let's select this RNA. So what I could do is I could go over here and I could just highlight it. And then I can do select RNA, comma, silly. So now this is defined on this selection. So as it's named it RNA. And so now if I click on this, it clicks on the RNA. And I can also do it through the selection. So like if I wanted to, let's select those isopropanol or those phenols. So if I do select, and then I put the name that I want to call it, so phenol, and then // C plus D. And so now it's going to select these. And so now let's color the phenol. So I could come over here and I could do color, and then it gives me a bunch of color options. Or I could do color, set gray, comma, phenol. Now let's color the RNA. So we'll do it this way. So we'll do color, and we'll do orange for this one. Orange, comma, RNA. Now you have that. So now we have the protein to deal with. So for the protein, if we go back to what we're trying to get, the protein, I have it colored by domain. So this is basically like the functional area or structural areas of the protein. And so I want to color it by that. So first I want to use some bases with stone to have base ladders. And I show cartoon. Sorry about that, people. Okay. So apparently when I had sidechain hopper on, it made the lines weird, so sorry about that. We're back. I was wondering what that happened about that. So I'll turn it off for now. Okay, so now we're going to try to set the domain. Now we're going to color based on the domains, which are like the functional or structural regions of a protein. And so I know there's one, since I want to color 1 to 140, it's the end domain, I want to color it a bluish color. And so I could come over here and select it all, and then it would be a selection, and I could name it as a selection, or I could like rename the selection. But instead I'm just going to do it the easier way and do it through this script. So what I'm going to do is select. So this tells me I'm going to select something, and then I'm going to name my selection in domain. Then //A to tell it it's in this like A segment. And then I'm going to also say and resi 1 through 140. And now it's defined this selection. So if I were to go over here and select it, it would be, oh sorry, if I were to go over to here and to press the end domain, it will have it selected. And so now what I want to do is color it. So I could come over here and I could do color and do it that way, or have a script that I wrote so I could do it for all of it at once. So I know that I want this sky blue, and so I can do, so sometimes I make the script where I do it over here, and then I just copy and paste. Or what you can do is you can actually like file, run script, and then you can select the script that you want to run. So now that's colored. So now I could do this for every different one of these, and so I'm just going to do that for the sake of time. just paste them all in here, and you can see that now I've created all of these different selections. And so now I could treat each of these selections like I can do things to it individually. But there are some things you can't do if it's just a selection. So these are all like still connected. So if you wanted to display like the surface, display surface. So if I wanted, it's been a while, sorry. Show surface. So now you can see the surface, display, so settings, surface, transparency, surface. I can set the transparency, so now you see that the surface over here, but I can't do that on the selection, like it has to be on the whole thing. So sometimes I will actually like create objects. So if you go to a selection, you can like copy the object or extract the object, and if something's an object, then you can do that individually. But we don't want to deal with that right now. It's just something to know. Okay, these are just some settings. So this, I like to turn the shadows off. So setting, I can do it.
Okay, so you did the ones that are like,
under like rendering, so like shadows I have none. See, but there's all sorts of different settings you can play around with. And then I don't like this like specular reflection where it's like glare. So I could do that, and then setting, where was it? Display, specular reflection. See, but you can see there's all sorts of different things. You can also, there's like a wizard, appearance, or no, not that. There's also demos. Here we go. So you can see like a demo of the different representations, and it shows you like different options. It's just demonstrations, and then end the demonstration. But basically, it's trying to show you the different things. Okay, now where did my protein go? Let's orient ourselves. Oh, so we're still in this mode. So let's go back to 4F3D. Okay, so now you can see that the view got all twisted. So before I had actually set the view. So let me just get back to where we were. And so I know this was back to where we were because when I was on it before, what I did was actually, if you do get view, now you can copy and paste this. Well, it copies it for you, and you can paste it somewhere to keep it, and that's going to keep the view. So if I go like this, and now I say get view, and now I go back and I copy that old one. See here. But if I had copied that other one, then I would have gotten to that same scene. And speaking of scenes, you can also save these different views and stuff as scenes. So you can go to scene, and you can add it this way. So you can like store it, and so these you can store it in like that function keys, or you can custom name them. So you do scene, then what you want to name it. So we're just gonna scene pretty store. And now we have a pretty, and then so say we want to do this, and then we can do like, oh yeah, so there's like, if you move your trackpad or your wheel or whatever, you can slice, so you just see certain regions of the protein. You can also do that with like display and clip or zoom. So if I wanted to like zoom in on this, so if I wanted to zoom in, good thing I can go back to where I was. So if you press the state button, it'll take you back to where you were at that when you save that state. Maybe we want to scene where we zoom in on the RNA. You can click action, zoom in. It's a little far. Let's go back a little so I can like pinch out. I'm using a trackpad, but mouse is better. You can like, there's different viewing options, I don't want it zoomed that much so I can play around. So when I'm doing the scrolling pad or whatever, it's dealing with this this clip. So it's like the clip is like the slab, so how much like thick of the thing you are around it. But anyway, so we're just gonna call this scene 3 end, because that's the 3-horn binding end. 3 end zoom store. Okay, so now if I go back, we have the pretty, and now we have the 3 end zoom. And so we could do all sorts of different things in these different states, and then always go back to them. So it's helpful when we like make movies and stuff, as well as just if there's some sort of representation that you don't want to lose. And so yeah, so there's a ton of different things. You really just have to play around. And so if you go to like edit all, you can see like so many different things, and you can also change them all in here as well, as well as do some more custom changes than you can with the user interface. Yeah, there's also a bunch of keyboard shortcuts, and you can make your own custom ones as well. Oh yeah, exporting them. So if I want to, you have this option of like draw or ray. So if you go over to here, you can see you can set the dimensions. You have draw, which is fast, but it's going to give you a lower quality one. Or you have ray, which is slower, but it'll give you a higher quality one. So if you're doing like a presentation or something, you want to use ray, but it's going to be really slow. But if it's just something like quick, you can just do this fast draw. And yeah, so another thing, so you can save your session with just like Control-Command S. When you working with PyMol, one thing you can do is you can, when you start a session, log file, if you like open, what this will do is it will like record what you're doing with when you're doing like the graphical user stuff, and then it'll make the scripted version so you can go back and see what it was actually doing, and then you could use it to help you write your own custom script that you can then run, and that sort of thing. So yeah, so that is all I want to show you today, and so happy playing.